All right, welcome back. We're out of the studio right now on the campus of Tel Aviv University, and this is the building I work in. Let's go inside, see how we're growing our plants, and talk a little bit about the real research that we're doing in the lab. Come on in. Okay, so now we're in the Manna Center for Plant Biosciences. This is the area in our department where we grow all our plants under various conditions. All of these rooms have literally hundreds of different types of plants growing under various conditions. Let's take a look and see how we grow the Arabidopsis. So what we have here actually are two different rooms, and we'll see them in 1 second. In this room, the plants are growing under a long day condition so that they'll flower quickly. And in this room, they're growing under a short day condition so that they'll take a longer time to flower. You remember this from the second lecture. We were talking about how the length of day can affect how a plant flowers. So in this room, what we have is literally hundreds of Arabidopsis plants growing. These, for example, are about 6-7 weeks old. They've been grown under a short day condition, which is why they have so many leaves growing on them. In a second, we'll go in another room and see what they look like under long day conditions where they flower faster. And under these controlled conditions, we get very reproducible results. What's great about these rooms is that we don't have to do anything to take care of the plants. These are automatically watered, automatically fertilized, and we can just check on them once every few days to make sure they're growing, and then come back and take the plants to our lab to do the various genetic or molecular work that we do on them. So these rooms are great for propagating hundreds of different Arabidopsis strains, for growing them to seeds, and for certain biochemical studies. But for exact environmental conditions that simulate changes in the environment, we use these very advanced growth chambers. And these growth chambers can be programmed to plus or minus 1/2 of a degree. And we can change the amount of humidity. And we can actually even change the color of light within it. So as you can see here in this chamber, these are some adult Arabidopsis. These are very close to flowering. And these have been grown under increasing temperatures so that we could see what would happen, for example, under global warming. Now I want to show you one of my favorite toys. As you remember, we learned in the second lecture about how plants grow under different colors of light, different qualities of light. Now, how could we do such experiments with white light all around us? Well, the way we carry out these experiments is we do them in a dark room, and there we have the different colors of light. So come on in with me. All right, so what we have here in this special chamber are lights of different wavelengths, LEDs. And here we can see one Arabidopsis plant that's been growing under far red light. And these are LEDs that are very high intensity, but it's about 720 nanometers. That's why you don't see it so bright, because it's sort of at the end of our visual spectrum. But with one push of a button, I could turn that now into turning on the blue lights. And now we could see what would be the difference, how this plant would respond in blue light. So here, there's very high blue light, high intensity blue light. Or, if we want, we could turn on red lights. And now actually you could see when the red lights are on, the far red lights look like they're off completely, so we can't even see the difference. So by changing how the red lights are on and how the far red lights are on, I can actually determine whether it's in the shade, whether it's high intensity light, whether it's in the morning, or whether it's just in a normal spectrum of red, blue, and far red together.